For decades, the standard clinical explanation for major depressive disorder has relied on the monoamine hypothesis, the assumption that depression is primarily a neurotransmitter signaling error, usually framed as a simple deficit of serotonin or dopamine. But this model fails to explain a specific clinical pattern: patients who show zero improvement after standard SSRI treatments. This resistance suggests a pathology that operates well outside the bounds of a chemical deficit. The focus shifted following a neuroimaging study involving over 1,000 patients. Researchers looked past receptor activity and measured the physical mass of the brain itself. The data revealed a physical, volumetric reduction of the hippocampus. In patients with major depressive disorder, this region is visibly smaller than in healthy controls. To understand why this volume is lost, we must deconstruct the depressed brain. We need to trace how unrelenting chronic stress breaks the body's endocrine system, which in turn prunes the architecture of the hippocampus. This evidence suggests that chronic depression may be driven by an active process of structural deterioration occurring within the brain's physical hardware. To see exactly how this hardware degrades, we have to scale down from the gross anatomy of the hippocampus and examine the microcircuitry of a specific area called the CA3 region. The macroscopic volume loss we see on an MRI is not caused by the mass death of entire cells. Rather, it is the result of extreme pruning. Individual neurons are dismantling their own communication structures. This is a healthy CA3 pyramidal neuron. Its apical dendrites form a branching network to process incoming signals. Under chronic stress, these branches wither, causing a 30% decrease in length and complexity. Zooming into a remaining branch, we find dendritic spines, critical connection points for memory storage. As branches retract, these spines vanish, driving a 25% drop in spine density. Without these physical structures, the brain cannot maintain basic cognitive processes. This precise structural decay explains the memory impairments and cognitive rigidness often observed in MDD. This brings up a mechanical question. What biological signal is instructing these neurons to dismantle their own receiving networks? The answer is not generated within the brain itself. The trigger originates deep in the body's peripheral endocrine system. The shrinking hippocampus is the cumulative result of millions of neurons simultaneously amputating their own connections. To find the source of that destructive signal, we must map the body's systemic stress response network, the hypothalamic, pituitary, adrenal, or HPA axis. In a healthy stress response, the hypothalamus releases CRH, prompting the pituitary gland to signal the adrenal cortex to release cortisol. Once the threat passes, a negative feedback loop takes over. Cortisol returns to the brain, signaling the hypothalamus to shut down CRH production. But continuous, unyielding psychological stress forces this system to operate outside its design parameters. Under constant threat, the neurons in the paraventricular nucleus of the hypothalamus become hypersensitized to stress inputs. They react more aggressively to every incoming stimulus. Flooded with constant cortisol, the brain adapts by developing glucocorticoid resistance. The receptors meant to catch the shutoff signal stop responding. The brain becomes deaf to cortisol's command to stop. The negative feedback loop shatters. Without that regulatory mechanism, the system is unmoored. The HPA axis defaults to pathological hypersecretion. It continuously dumps elevated levels of both CRH and cortisol into the bloodstream. These stress hormones evolved for brief, intense bursts to survive acute danger. When they circulate constantly, they become toxic to the tissues they were designed to protect. Chronic stress breaks the body's primary regulatory thermostat. It locks the endocrine system into a state of perpetual overdrive, manufacturing the exact chemicals that will tear the brain apart. This endocrine overdrive does not stay confined to the bloodstream. Sustained HPA hyperactivation triggers an aggressive immune response, heavily upregulating systemic pro-inflammatory cytokines like TNF-alpha. Circulating TNF-alpha attacks the blood-brain barrier. It violently pulls apart the physical seals between endothelial cells, creating wide gaps in the brain's primary defense system. With the barrier breached, peripheral inflammation floods into the central nervous system. This intrusion activates microglia, the brain's resident immune cells, causing them to continuously release destructive reactive oxygen species. Psychological stress is not contained to the mind. The body's reaction actively burns through the brain's physical defenses to establish a localized state of chronic neuroinflammation. Now we can return to the CA3 dendrites to understand exactly how this toxic, inflamed environment executes physical pruning. The destruction relies on a two-front assault. The first weapon is excitotoxicity, driven by the massive excess of glucocorticoids entering the hippocampus. In this synapse schematic, glucocorticoids force local mossy fibers to release glutamate in concentrations that overwhelm the synaptic cleft. The neurotransmitter spills over, hitting extrasynaptic NMDA receptors located outside the normal signaling area. Activating these extrasynaptic receptors forces an influx of calcium into the cell. This triggers calpain, a destructive enzyme that cleaves the neuron's internal proteins, causing severe cellular damage. While excitotoxicity burns the neuron from the outside, the second weapon attacks from within, the direct toxicity of the hormone CRH. The elevated levels of CRH generated by the broken HPA axis bind directly to CRFR1 receptors located right on the dendritic spines. This binding blocks the polymerization of F-actin. Since F-actin is the structural protein holding the spine's physical shape together, the spine simply collapses. The combination of excitotoxicity burning the neurons from the outside and CRH dismantling their internal scaffolding forces the dendrites to rapidly retract to survive. Usually, the brain can recover from damage. To see why it fails here, we have to shift to the subgranular zone, the brain's primary repair center located in the dentate gyrus. In a healthy system, adult neurogenesis occurs here constantly. Neural stem cells divide and differentiate into new neurons to repair damaged circuits and help regulate mood. This line graph tracks brain-derived neurotrophic factor, or BDNF, the critical fertilizer allowing new stem cells to grow. Under chronic stress, BDNF levels plummet by 50%. Stress hormones cause an epigenetic silencing of BDNF promoters. Stripped of survival signals, the newly formed stem cells halt their growth and die before integrating. Chronic stress destroys existing neural architecture while simultaneously disabling the biological mechanisms intended to repair it. By zooming out from the cellular level, the full scope of the disease becomes clear. We can now summarize the complete interconnected collapse. HPA axis hyperactivation drives a flood of toxic hormones. This barrage breaches the blood-brain barrier, ignites neuroinflammation, executes the structural pruning of the CA3 region, and halts noogenesis in the dentate gyrus. The clinical symptoms of major depressive disorder are the direct output of this missing neural hardware. Memory loss, emotional blunting, and cognitive rigidness are the behavioral results of a physically shrinking hippocampus. This biological reality demands a shift in how we approach psychiatric treatment. Attempting to simply boost serotonin levels in a brain that is actively being dismantled by inflammation and endocrine toxicity ignores the root physical damage. The future of treating chronic stress-associated depression relies on protecting the brain's physical architecture. To stop the brain from shrinking, we have to extinguish the neuroinflammatory fire and restore the balance of the HPA axis.